Dr. Anita Koshy is an associate professor in the Department of Neurology and the Department of Immunobiology at the University of Arizona. Dr. Koshy studied biology at Stanford, where she first discovered her love of neuroscience research. Dr. Koshy received her medical degree from Duke University, where she confirmed her desire to be a clinician scientist. She later specialized in the area of infectious diseases. During her postdoctoral fellowship at Stanford, Dr. Koshy established a technique to identify which cells in a mouse brain have interacted with the parasite Toxoplasma gondii. In 2012, Dr. Koshy moved to the University of Arizona to establish her own lab, which continues to pursue understanding the molecular underpinnings of the brain-toxoplasma interaction. In my interview with Dr. Koshy, we discussed the effects of toxoplasma on host behavior, the ethical considerations involved when studying the human brain, and her vision for the future of artificial intelligence in neurology.
My name is Anita Koshy. I am a physician scientist, and which means that for me, it means that I see patients relatively rarely, and then for most of my time, I am, as my kids know me, a mouse doctor. So I study how a parasite is able to persist in the mammalian brain. That parasite is called Toxoplasma gondii.
And can this parasite also exist in the human brain?
Mm-hmm. So we think that someplace between an eighth of the world to a third of the world is infected with Toxoplasma, and once you're infected, we think you have the parasite persist in a variety of organs, including the brain.
So what is your research looking for exactly?
So we're trying to understand, so for most people who have the parasite, they have it, they've had it for some number of time, and they have no symptoms from it. Mm-hmm. But if they have that persistent parasite, and then they develop an immunocompromised state that's very serious, like AIDS or a bone marrow transplant, that persistent parasite can reactivate and cause problems. And so our current therapies are targeted at sort of the fast replicating form of the parasite, but not at the form that persists in tissue. And so the idea is that if we can understand how or why the parasite is persisting in the brain, then maybe you can design drugs that target that.
So the parasite is typically confined to the brain area?
It is, in humans, brain, skeletal muscle, and cardiac muscles are the best documented places in sort of the chronic phase.
So why are you looking at the brain in particular?
The brain, because it is the place that seems to have the highest burden of the parasite, and it is where, when people are symptomatic, it is where they are most symptomatic usually.
What do the symptoms include?
They are generally focal neurologic symptoms, so the parasite will be in a very specific part of the brain and affect the function of that part of the brain, and therefore you'll get symptoms related to that.
So does this parasite affect the human behavior or the mouse behavior that you've seen?
There are a number of studies that suggest that the persistent form of the parasite affects rodent behavior. That's been done by multiple different labs in different paradigms. There are also some negative data, meaning that people infected the mice or the rats with the parasite and then didn't see an effect. Mm-hmm. But there have been a number of studies that suggest there are effects. The human data is interesting. So in the mouse or rodent model, you know exactly who's getting infected, how long that mouse is infected, things like that. In human data, you're relying on serology. So serum tests or blood tests that say, yes, this person is infected with toxo or no. In human studies, when you're dependent on serology, what you don't know is the behavior that you're studying more likely to cause you to be toxo positive, like it puts you at risk for eating undercooked meat, drinking water that's not clean, things like that, sure. Versus is it having an infection that's causing the behavior. So it's very much a chicken and egg issue. And one of the more interesting studies is that one group used mice that cause a persistent infection, or not mice, parasites that cause a persistent infection, and then parasites that don't cause a persistent infection. And the most well-known behavior that toxo causes is this decreased aversion to cat urine. So mice should be afraid of cat urine, or rats should be afraid of cat urine, and toxo-infected mice show less they go into the quadrant that has the cat urine more frequently than they should. And what was interesting about this one study is most people have linked that behavior to where these cysts persist or where the parasite persists in the brain.
Sure.
But the study that used a non-persistent form of the parasite also found the behavior. And so that's interesting because it says, well, maybe it has nothing to do with where the parasite goes and stays. Maybe it has to do with the amount of inflammation you get or where you get that inflammation. Right, and that's very easy to sort of understand.
Yeah, so in this sense, do you feel like it might be sort of like an evolutionary mechanism to, continue up the food chain for the organism, or do you think it's just kind of a secondhand detrimental effect on the host?
I think that because toxo infects so many animals, right, so everything from birds to humans, so there are plenty of ways for the parasite to get back into its definitive host, the cat.
Sure.
So I don't know that I think this is an evolutionarily derived, really driving force. It's not like some of these really elegant systems where they've shown, that ant has to crawl up here
right,
and they
the
get picked up.
caterpillar
Right, yeah.
lifts up or
right,
something.
exactly.
Yeah.
Like, those are where you can really say, like, well, there's no other way for the parasite to get back to its definitive host. While with Toxoplasma, there are plenty of ways for it to get back to its definitive host or even spread from intermediate host to intermediate host. So humans often get it from an intermediate host, and so undercooked meat, pork, lamb, things like that.
Sure.
And so then I think there should be, in theory, less evolutionary pressure on specifically causing mouse behavior or rodent behavior. It should be noticed that in one of the studies that's very famous for this aversion to cat urine, when they took a towel that had been rubbed on a cat, so a much stronger signal that the cat is in the presence of, is in that area, then the Toxoplasma infected rodents behave just like the non-infected rodents. And so that there's certainly some subtlety there in how that's manipulated or mediated. I don't know.
Yeah. Do any ethical issues come up in your work?
I think that ethical issues come up on some level with this idea of could the parasite be causing human behavior changes, and the ethical issue there is primarily the idea of let's just be sure we all understand what the data suggests and what it shows in terms of cause and effect. And the reason for me that's an ethical issue is because I have a sense of how much mental illness can be very hard for a patient and a family. We don't do a great job of treating mental illness generally. We don't have great, there's no quick pill often that fixes things. And so then people are trying to search for answers. And so I think it behooves us to be very careful about suggesting that the answer is that you have a parasite in your brain and whether or not that parasite in your brain, treating it would change anything or not. So I think that for me is that fine line to walk. And it's not to say that for any given person, having inflammation in your brain, if you were already genetically predisposed one way or the other, couldn't be the thing that tipped you over, right? But I think we should be careful about thinking about it and trying to apply it broadly and things like that.
How do computers factor into your research, if at all?
One of my grad students, Oscar, set up a system to scan brain sections and identify where very specific types of neurons that have interacted with the parasite are. And then he could set up the system to find them, count them, and then he could go back and manually, he and an undergrad, Colin Potter, could go back and manually say yes and make sure those was counting the right thing.
Sure. Yeah.
But that's the kind of thing that to do one mouse well is unbelievably painful. But if you can do it well so that now for your next 7 mice, it's pretty much automated and you're just checking, click, 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 it's amazing.
Do you think there will ever be a time when you won't need an undergrad to fact check what the computer is doing?
I can imagine that there would be a time that it's hard for me to imagine that there's not always going to be a time where you just want to verify because it's it's garbage in, garbage out, right? And so people talk about about these systems where automated interactive systems that became racist because what they were getting in, or right, or these biases. And so you can imagine that then you need a check on what have I fed it that it thinks it's learned or hasn't learned.
I think it's easy for a lot of people to picture the bias that's possible in that sort of algorithm in sort of a social stance, so like for race or gender or some sort of profiling. But how do you see that maybe coming about in science?
I know that's harder because we always think of ourselves as so unbiased. But you know, it's one of these things where, so I'll give you a simple example that is very silly, but is sort of simple at the same time, but I think illustrative, which is that when I was working with a friend of mine, Mary Buckwalter, we were working on a program, and we were basically counting immune cells, and there were mice that had a modified transgene, basically a modified gene, versus mice that didn't, and they were all infected with parasites. And Marion very smartly said, look, when the tech or whoever's going to analyze these slides, the grad student looks at them, we should make sure they don't know which mouse is which, right? Yeah. So let's just make sure they don't know, which is actually much more common now, but wasn't particularly common. So you blind the scientist, right? And I don't think anyone would have intentionally, right, said, okay, well, I know this mouse is this, and so let me count these, but you might, in ways you don't understand, accidentally bias yourself, right? Say like, oh, well, look, there's a big clump of immune cells, I'll count those. But for that, I don't see so many, and so on some level, people would argue that computers wouldn't do that, but I just anticipate that there are ways in which you could accidentally set up a system where you bias it in an accidental. And the sort of hard part about that is because we think of it as unbiased, we might be less willing to question the data, right? So we might say, like, well, that's what it shows, like the computer did it, it wasn't me, so it must be correct. I think there's massive amounts of information that will be gained by automating things, and just like the sheer numbers, right? So the human, like having to tie someone to their lab bench to like count 1,000 parasites or whatever is going to be much harder than having a program that does it, right? But I think I'm a humanist, so I think we matter, right? So I think that like it's good to have people who are there to check it and to think about it, right? Well, what does this data mean? Does it really mean what I think it means, or, right? And there was just a debate, I think, at one of the neurology conferences that I was not at, about whether or not neurologists would get replaced by AI, right? And there were two sides, and people were arguing about it. But it is, for me, especially neurology, which is all about the exam and how well do you do the exam. That is what helps decide, is it in this part of the brain, is it this part of the nervous system, is it in the muscle, whatever. It is hard at the moment for me to think how you could automate that, where you have someone who doesn't know how to think through this, just putting in whatever they think is right, because if it's garbage that you're putting into it, again, whatever you get out is going to be not very good. And at the same time, at least at the moment, I can't imagine anyone being diagnosed with something like Alzheimer's, ALS, these brain tumor, right, without having a human to help them talk through that. At least for now, I think that we won't have computers.